Okay, by now we are quite aware that what is kingdom Monera all about, what is a bacteria, it is a living organism, what kingdom Monera prokaryotic cells are all about. Now what we have to understand next is acellular type of organisms. Now when I say acellular, I mean the organization where the organism is not having the cellular level of organization inside it. Now what was cellular level of organization? It referred to an organization where the organism had a well defined nucleus which inside it had the genetic content and rest of the organelles. Now if it had the organelles which were enclosed by a double membrane, it happened to be a eukaryotic cell. If it didn't have that, it was a prokaryotic cell. Now there are certain cases, there are certain organisms in the evolutionary history, if we specifically see, if we find acellular organisms, and one organism that is of great interest to many of our microbiologists are the viruses. Now you all must have heard about Louis Pasteur. He was the one who pioneered in the study of microbes basically. Today whatever study we are doing in the microbiology field, he was basically father of microbiology because he started studying about rabies, and while studying about certain microbes, he just coined out that there were certain microbes which could not be seen even under the microscope. Though it didn't come to his notice that later on electron microscopes and rest of the microscopy would get so advanced that we will be able to see many small particles under the microscope, but at his time what he studied is that the causative agent of rabies, that is the rabies virus, he could not see it, and he said it that it was the causative agent and it was something which could not be seen under the microscope. Later on it was seen that the causative agent of certain diseases, they were quite smaller in size, that is around 10 to 200 nanometers, so small in size, and it came to their notice that they were acellular organisms. Now yet again I start and come to the same point from where I started, that was acellular organisms. Now why is a virus classified as an acellular organism? The reason is that the virus is not having a cellular level of organization. All it has is a genetic material which could be DNA or it could be RNA, and it has a protein coat. So basically if we talk about a virus, it would be a virion, that would be the genetic content, and a protein coat which is surrounding. Another thing about viruses is that they are living inside a living cell only. Okay, one by one we are going to see how they were discovered, and we are going to learn by heart the name of few scientists who were important. As I told you, Louis Pasteur was the one who started it. Later on there was a scientist known as DJ Ivanovsky. He was a Russian scientist. He saw tobacco mosaic virus. I have mentioned over here TMV, it is tobacco mosaic virus. You know, tobacco is an important crop which is a cash crop, though it leads to certain diseases, but still it is unfortunately an important cash crop, and lot of study is invested in this plant. So tobacco mosaic virus was a virus which caused a disease in tobacco plants, and this scientist, this particular scientist, he saw this, that these tobacco mosaic virus is some causative agent. He just figured it out that there is some causative agent which is leading to a diseased condition in tobacco. Later on, like after few years, 4 or 5 years, there was a Dutch scientist, that was Mr. M. Bedring. He said that whatever is the causative agent, whatever happens to be the causative agent, it is not bacteria first of all. Secondly, it is a liquid form of infectious agent which is present only in the living cells. It is not present in the cells which are dead. It happens to be present in the living cells, and he gave this term, that is contagium vivum fluidum, that a natured causative agent is responsible for causing the disease. And one thing he could distinguish, that the living content, it is this particular causative agent of the disease. Again, it was TMV which was being studied, tobacco mosaic virus. Bacteriophages were not studied till then. Now what are bacteriophages? We are going to come to that. But the TMV was believed that it is a liquid state of infectious fluid which goes from living cells, and it stays in case the fluid is present in the living cells only. It won't be present in non-living cells. Now what is is so strange about viruses is that first of all they are obligate parasites. That means they cannot survive without a parasitic behavior of nutrition. That means they are going to infect some or other cell. Being so small in size, they can even infect the bacteria, they can infect all the organisms of all the kingdoms whichever are present. So we find viruses which infect bacteria, we find viruses which infect protistants, we are going to find viruses which are going to infect plants, animals and rest all organisms, as I told you. All the organisms whichever are present in all the kingdoms, they can be infected by viruses. And as I told you, I mentioned the name bacteriophage. Bacteriophage is a virus which infects the bacteria. Now, moving further, later on there was a he was American microbiologist, Wendell Stanley. What he said was that the TMV that was studied by these two, it was not liquid, it was not bacteria, it was a virus which was of particulate nature. Okay, this person was responsible for giving the term virus, and this particular person, Wendell Stanley, he was responsible for the particulate nature of telling that the TMV is of particulate nature and it has particle form, and basically he is being given the credit of the structure of virus, which says that virus is virion plus the protein coat. So let us see what viruses all are about. Enough of introduction has been there. Now what you see over here is, it appears like some sort of gadget of a villainous sci-fi movie. You would see, might have seen in movies that the villain or the hero, he uses such sort of sci-fi gadgets in their use, and this is not that. First of all, this is a virus. This is a bacteriophage virus which would go and infect the bacteria, and we know that bacteria are highly notorious. They are responsible for giving us diseases. These viruses, they give diseases to bacteria. So this is the typical structure of bacteria. I've used this just to make you little bit interested in what virus and virology would all be about. First thing that we have is, what is the structure and function of viruses. We are going to understand the structure over here. They are simple structures. They are living in the cell, in the living cell, and they are non-living. As I told you, they are obligate parasites. They cannot survive without a living cell. So what would happen to them if they are not having a living cell? They would be dead. Okay. So viruses are simple structures which consist of capsid. Now as I told you, they have a protein coat and a genetic material. So that protein coat is known as capsid. The name for that protein coat is capsid. A protein coat made of individual protein subunits which are known as capsomeres. So we have a capsid, that is the protein coat. The protein coat has individual protein units which are capsomeres. It provides the virus with its shapes, make up 95% of the virus size. Now come back to what was the introductory picture. This happens to be the protein coat, okay. This outside peripheral boundary would be the protein coat, and inside it there would be a genetic material. Now the genetic material could be single stranded DNA, it could be double stranded DNA, it could be single stranded RNA, it could be double stranded RNA. It depends on what type of virus it is. Genetic material is located inside the capsid, that is the protein coat, and it could be DNA or RNA, and depending on the strands, number of strands, it could be single stranded or double stranded. Now come to the structure of capsid. As I told you, it has capsomeres, that are individual units which are present. So this is the capsid. It is a protein coat. It encloses and protects the nucleic acid of the virus. Now that nucleic acid is known as virion, okay. So it accounts for most of the viral mass, as we studied, as much as 95%. It is composed of single or multiple proteins. A single subunit is called capsomeres, and you must remember that these capsomeres are often formed of proteins, and certain glycoproteins are also present, and they are responsible for infection caused by virus. So they assist in infection that is to be caused by virus. Over here you can see there is a nucleic acid inside, there are capsomeres, and this is the simplest representation of the capsid. Next what we see is that the viral capsid, It is surrounded by protein coat. Again, it is repetition. It is of three types. The types which are based on the shape that the capsid is having. First is helical, that the capsomeres would be arranged in the helical pattern, that is seen in the case of tobacco mosaic virus, the first virus that was studied, TMV. Then we have icosahedral shape, that is there are 20 faces. Like we have polyhedron, this icosahedron has 20 faces, and there would be 20 faces of the protein coat, and that would form the proper capsid. And last one is complex, it is not icosahedral, it won't be helical. The capsomeres would be arranged in a complex pattern depending upon the type of the virus that would be present. Now what we have next is, we are going to study about two types of viruses. First is bacteriophage, we have to understand its structure, and we have to understand the structure of tobacco mosaic virus as well. Now as you can see, this bacteriophage belongs to that icosahedral shape, or we can say polyhedron is there. The capsid is the head, this is the protein coat, it is around the nucleic acid which is present inside this capsid, that would be the genetic material. Capsid is divided into subunits that are called capsomeres, that is in the case of bacteriophage. The body is going to be the central rod shaped structure, has retractable sheath. So you see, you saw in that picture, that first picture, we had this head and body and tail. These are the three parts of the bacteriophage. Now inside this rod shape there is a hollow core, because the nucleic acid is confined to this part. Now why does it have this rod? Because the infectious part, from where it is going to enter into the bacteria for infection, that is through tail. So the connection between the nucleic acid which has to go inside the bacteria after infection and between the infectious tails happens to be the rod. Okay, now at the end of the core is the tail, 6 tail fibers which help to anchor the virus to its host, and as I told you, it would be glycoproteins which would be helping in infection. So this is about bacteriophage. Next what we have is the structure of tobacco mosaic virus. Tobacco mosaic virus, as I told you, it is a helical structure, the capsid is helical, and as you can see over here, it is having lot many capsomeres. It appears like some sort of leafy arrangement, but it is not leafy. These capsomeres are proteins, and inside it you can see the helical RNA as well. The capsomeres are also arranged in a helical pattern, and what you have to remember is that the core is hollow. The diameters are to be learnt by heart, that the diameter of this entire protein coat of TMV is 180 angstrom, and the radius happens to be 40 angstrom of the hollow core that we are talking about. The pitch of each helix. Now what is the pitch? Pitch is after how much angle or how much degree it is going to take the turn. Supposedly you are climbing the stairs which are helical. After how much time an entire helix is getting completed, that is known as pitch, and the pitch happens to be 23 angstrom. So this is about the structure of tobacco mosaic virus. So you see, this is what a virus looks like, and we have had enough knowledge what viruses are all about. Next we are going to get introduced to a term that is viroid. Last one, and with that we are almost done with these acellular particles. Okay, what are viroids? As I told you, virus is having a protein coat and it is having a genetic material. In case of viroids, one thing would be missing, that is there won't be any protein coat. They are the smallest particles, smallest infectious agents. First of all, in around 1971, T.O. Diner was the scientist who discovered a potato spindle tuber disease causative agent. There is a disease known as potato spindle tuber disease. The causative agent for that particular disease was nothing but an RNA strand basically. The RNA was infecting the particular plant, and it was causing the disease in the plant. This particular RNA happens to be viroid, and it is not having a protein coat. So you see that it would be non-living outside. When it goes inside, it is going to cause some sort of infection, and there would be transcription and reverse transcription, and then there would be formation of proteins and the disease would be caused. So you have to remember that where it was discovered in potato spindle tuber disease causative agent, and it is nothing but a but an RNA basically that causes the infection, so it would be the smallest particle. Apart from these viroids, there are prions, which are proteins which cause disease, and they are smallest because you see there won't be any protein coat, not any protein coat required. You have to remember the scientist's name, you have to remember the disease that they cause and from where they got famous, and what are they all about. Nothing but only an infectious RNA that would go and integrate inside the cell. It would go inside the cell, it would perform the functions which would be hazardous to the plant. So this is all about the acellular organisms that we study, which are not under classification of any 5 kingdoms, whether Monera, Protista, they are not classified under any of the kingdoms, but yes, they are to be studied with Monera itself, because when we study about Monera, the study of viruses almost started with considering that they are some sort of bacteria, but they were not bacteria, and they are simple viruses. They cause lots of diseases, and none of the viruses is going to be that or which would give you any sort of economic importance. That would be, as is seen in the case of bacteria, there are certain bacteria which help you in a positive way, but none of the viruses is going to help you until and unless you learn about their evolutionary history and use them to certain aspect in the biotechnological fields.